Hi everyone, this is Dr. Prityavari. We'll begin with microscopic structure of a compact bone. Now, this slide is called as ground section of compact bone. We are going to see the longitudinal section and transverse section together. Now, why we call it as ground section? Generally, this slide is made from a smallest bone as phalanx, where you need to grind it actually so that you can see a very thin sheath of bone which is as good as transparent, and then it is mounted and we make a slide from it without staining. So you can make out in this slide or in this diagram, nothing is stained here. We are not using H&E stain here. So in the under the microscope, when you see this bone, you can see very pale yellow colored bone, and in between the pale yellow colored calcified matrix you can see there will be spaces. So wherever there will be a space for passage of blood vessels, nerves, lymphatics, or for the cells, that will appear empty, that looks transparent white in color. You need to understand this. So under microscope it is very easy to identify this slide, and you need to close the window of your condenser a little bit and adjust the light source, then you can see this slide clearly. You can see most of the features under low power, as if in this picture, whatever is seen can be seen under low power. So, what is compact bone basically? Compact bone is the shaft of the long bone. When you see the diaphysis, metaphysis, underneath the periosteum, that bone is called as compact bone, which is surrounding the bone marrow cavity. And it has got a peculiar arrangement, very good structure, very nicely arranged lamellae together form the structural and functional unit of a compact bone called as osteon or Haversian system. Now we all know the long bones are covered by periosteum. So let us begin from outside. We'll go from outside inside inward. So even we can make out the periosteum under microscope. A thin sheath of collagen fibers can be seen. So outer layer of fibers, and inside there will be a layer of cells. These are, you can say, kind of stem cells, osteoprogenitor cells, which are lining the periosteum. So periosteum is required for regeneration of a bone. Suppose there is a injury, there is a break, fracture in the bone, and wherever remodeling of the bone is taking place, there you need osteoprogenitor cells. So if the periosteum is there, then it can lay down the bone over there. So osteoprogenitor cells get converted into osteoblasts. Osteoblasts then differentiate into osteocytes. So here in this diagram, you can make out two layers in the periosteum, outer fibrous, inner cellular layer. Now, inside, what is the basic structural and functional unit will be the Haversian system or osteon. So you can see in the center there is a canal, a rounded canal. These are the elongated canals which are surrounded by sheets of laid down bony matrix called as lamellae. So here this is one lamina, this is second lamina, this is third lamina. This way you can see many lamellae within one Haversian system. So what are laminae? These are the bony matrix laid down by the osteocytes which are entrapped in between. So in between the lamellae you can see here rounded black color lines. Actually these won't appear black, they will appear transparent. And in between you can see there are spaces. These spaces are lacunae for the cell's osteocytes. So this complete structure is called as osteon or Haversian system. And inside, again, there will be bone marrow. So that bone marrow will also be lined by endosteum, where again the lining will be formed by osteoprogenitor cells. So this is a basic structure if you see in a compact bone. We are going to see the details afterwards. So in the same diagram, you can say it is a 3D diagram. So you can see the uppermost part is the transverse section and this lower part is the longitudinal section. So we are going to see both of them together. Okay. So we just understood what is periosteum, endosteum, and what is Haversian system. Now we'll see details of the Haversian system. The Haversian system has got Haversian canal which are placed longitudinally. So in the LS you can see the Haversian canal and in the TS they appear circular. They are surrounded by concentric lamellae. There are circularly arranged lamellae. In between the lamellae you can see spaces called as lacunae. So this is one lacuna, this is another lacuna. This is a space for accommodation of the osteocyte. Osteocytes are entrapped in this space. Now, this lacuna gives out small processes which are shown here with the black color, but actually again it won't be black, it will be white in color, transparent. These are called as canaliculi. These are for the cytoplasmic processes of the osteocyte. Now, we haven't processed this slide, we just grinded the phalanx to form this slide. So, try to understand. In the slide, you cannot see osteocyte. The osteocytes are dead already, so you can't see the osteocytes here. They are being washed off, you can say, at the time of processing. So what you can see is empty space. That empty space denotes that osteocyte were present in the live bone here. So this is a lacuna. So there are many lacunae present in between the lamellae. And from the lacunae, the canaliculi are coming out. Canaliculi are for the cytoplasmic processes of the osteocytes. Now I am introducing one more term here, that is Volkmann's canal. Now in the longitudinal section you can nicely see the two Haversian canals are connected to each other by Volkmann's canal. As you know, Haversian canals are for the passage of the neurovascular bundle, you can say, the blood vessels, nerves, lymphatics. Now what is the function of the Volkmann's canal? Volkmann canal is going to connect the adjacent Haversian canals, and it will also be a medium. It will act as a canal again for the passage of the blood vessels, nerves, lymphatic from one Haversian system to another Haversian system. Volkmann canals also connect the periosteum with the endosteum. Now this is a high power view of osteon. I want you to see the lacunae clearly. Now, just for the sake of understanding, I have shown the blood vessels between the two Haversian canals. You can see here connecting two Haversian canals, there is a Volkmann canal, and in the Volkmann canal, the nerves, blood vessel, lymphatics are shown. But you can't see this under microscope. Okay, this is just for the sake of understanding. I drew this diagram. In the lacuna, you can see a osteocyte. See how the osteocyte is giving out cytoplasmic processes, and the canaliculi of two lacunae, they communicate with each other so that the cytoplasmic processes will form cell junction, and they will also communicate with each other, and they can pass nutrients to each other. So this cytoplasmic processes and canaliculi, their purpose is transfer of nutrients and metabolic waste products from one cell to the adjacent cell. A very beautiful diagram. You can see the concentric laminae here. So this is a high power view. Under 40x, you can see the canaliculi very clearly. Now let us talk about few words about the lamellae. Lamellae, what are lamellae? These are the thin sheets of bony matrix laid down concentrically by osteocytes around the Haversian canal. Now, in this picture, you can see around the Haversian canal, these are concentrically arranged lamellae. So, these are the first type, concentric lamellae or Haversian lamellae. Second type is just after the endosteal layer, you can see around the central bone marrow, concentrically arranged lamellae. These are called as inner circumferential lamellae. They will be present throughout, encircling the bone marrow cavity, just after the endosteal layer. Same way, underneath the periosteum, surrounding the complete diaphysis, you can say, there will be 3 to 4 or maybe more layers of lamellae, which are circumferentially arranged, called as outer circumferential lamellae. Now, as the bone grows, and with the use of various joints and muscles, which are applying forces on the bone at various regions, continuous remodeling of the bone takes place. So, new osteons will be formed and the old osteons will be destroyed. So, in between the osteons, we can see some parts of old osteons. We call them as interstitial lamellae. So, there are 4 types of lamellae: inner circumferential, outer circumferential, interstitial, and concentric. Bone matrix in the lamellae, it is made up of inorganic and organic component. The basic inorganic component will be hydroxyapatite crystal, calcium phosphate. Organic component will be type 1 collagen fibers, ground substance which has got glucosaminoglycans, proteoglycans. Adjacent osteons are separated by a cement line which are devoid of collagen fibers. Sometimes you can see the cement line in the longitudinal section. Now, few words about the longitudinal section. This is a longitudinal section of a compact bone. We have many beautiful slides. Even in your lab, there will be slides which will be showing longitudinal section. In that longitudinal section, you can see the lamellae are longitudinally arranged around the Haversian canal. So, longitudinally running Haversian canals can be seen. You can see complete Haversian system surrounded by cement line. And at few places, you can also recognize Volkmann's canal if it is present. So here the lamellae will not be seen circularly, they will be seen parallelly arranged. And how are the osteocytes? Osteocytes are not circularly arranged here. Osteocytes are also longitudinally arranged in the lacunae. So lacunae are longitudinally placed over here. This is the longitudinal section. Now, what are various types of cells present in a bone? The first type of cell is osteoprogenitor cell, which is located in the cellular layer of periosteum and in endosteal layer. They give rise to osteoblast. The osteoblasts are bone forming cells which will synthesize organic component of bone matrix. Then osteoblasts give rise to osteocytes. What are osteocytes? These are derived from osteoblast. These are the mature bone cell present in lacunae. They will maintain the matrix. What are osteoclasts? These are multinucleated cells. They are involved in bone resorption. So whenever bone remodeling it taking place, or suppose there is a fracture, then first of all you need to clear that area. That is done by bone resorption by osteoclast. They are present in lacunae called as Hoship's lacunae. These are multinucleated cells. Let me tell you clearly, you can't see these cells in a ground section of compact bone. Few words about ossification of a bone. Bone can be ossified or bone can be formed in 2 ways: enchondral ossification and intramembranous ossification. In enchondral ossification, first there will be formation of a cartilaginous model of hyaline cartilage costal variety, and on top of that model, the bone formation takes place. So you can say the chondrocytes are being replaced by the bone forming cells, and the complete cartilaginous model will be replaced by the compact bone. Intramembranous ossification is generally seen into the flat bones as ribs and the flat bones covering the brain box. That's the squamous part of temporal bone, frontal bone, parietal bone, squamous part of occipital bone. So what happens here? First of all, a flat mesenchymal sheath will be formed, and in that sheath, ossification takes place. This gives rise to a flat bone having inner table, outer table, and diploid in between. This is just to simplify and introduce what is ossification, enchondral or intramembranous ossification. Please subscribe, share, and like. Thank you for watching.